Welcome to the first part of our notes about blood and blood spatter. You can see on your screen what you'll be learning how to do across these two sets of notes. So first, we have some genetic terms to remember. We have phenotype, a set of observable characteristics of an individual. You might remember this as phenotype physical because it is the physical trait that we see. This is compared to genotype, which is the genetic constitution of an individual, genotype genetic. You may also remember the term homozygous, having two of the same gene, the prefix homo meaning same. This could be either dominant or recessive. And then heterozygous, the prefix hetero meaning opposite, having one dominant and one recessive copy of a gene. Now a little bit of an introduction and history for you. So blood typing can provide us with class evidence, whereas DNA profiling can provide us with individual evidence. So we can typically get a DNA profile from blood evidence. Now blood spatter, something you're going to learn about in a later video, can give us information about the truthfulness of an account of a witness or suspect. Blood spatter can also provide information about the origin of the blood, the angle and velocity of impact, and the type of weapon used. All of this would be beneficial in figuring out what happened or proving what happened at a crime. Now, when it comes to the crime scene investigation involving blood, first step is going to be for search for blood evidence. I think that makes a lot of sense. Now, you may be thinking, isn't it obvious? Blood is bright red. But sometimes our blood can be very small and have gotten in very inconspicuous locations, so you do want to perform a thorough search because blood is really good evidence for us. If you do discover any blood, the first thing you want to do is determine whether the evidence is actually blood. A lot of things can look like blood that aren't blood, like ketchup or cranberry juice, things that have a red color could mislead investigators. So we want to perform what is called the Kastle-Meyer test, which you will be performing this in class. The Kastle-Meyer test allows us to determine whether something is blood or isn't blood. Then we're going to want to determine whether that blood is human, and then we are going to want to determine the blood type. We'll be talking about whether the blood is human and the blood type more in this video. Once we have done that, we're going to interpret our findings. We're going to see if the blood type matches a suspect. If it does not, we're going to exclude that suspect because if they don't have the same blood type, they aren't going to have the same DNA. And if the blood type does match, we're probably going to move on to DNA profiling to confirm that it is a match because like we said, blood typing is only class evidence, while DNA is going to be individual evidence. Now regarding the human or animal blood, the Kastle-Meyer test only tells us whether blood is blood or not. Any animal blood would be determined as blood. So we do need to determine whether it's human or animal because there are cases that animal blood could end up at a crime scene either from an animal being hurt as part of the attack or on accident. One of my dogs clipped my other dog's ear the other day and ended up bleeding a little bit while they were play fighting. And that got blood for a completely innocent reason at my house. So we perform what is called the Praecipitan test that it allows us to determine whether blood is human or animal. Now, blood is not just a red liquid. It has a lot of components to it. So whole blood, what you're thinking of, is made up of cells and plasma. Now, plasma is actually the fluid component of blood, and this fluid is going to contain hormones, clotting factors, as well as nutrients. Now, plasma is actually almost clear. It tends to have a yellowish tint to it, but it's pretty clear in a healthy individual. So whole blood, you're used to it being red, so why is it red if plasma is mainly clear? And that's because of red blood cells. Red blood cells carry oxygen to the body cells and carbon dioxide away. Now, a little rumor that I do like to dispel is that red blood cells, you've probably been told, turn red when they have oxygen and turn purple as they give their oxygen away. And this is technically true. However, In general, our red blood cells, as they make it through our body, still have oxygen left by the time they make it back to our lungs again, so they never actually turn blue or purple inside our body in a healthy individual. They can if something's wrong, but in general, your red blood cells are always red. Then you also have your white blood cells. These fight disease and foreign invaders. And this is actually why our blood contains DNA, is because of our white blood cells. Red blood cells, interestingly, do not contain any nuclei and therefore do not contain any nuclear DNA. However, white blood cells do have a cell nuclei, and therefore that is where our DNA comes from. When we say we test blood for DNA, we're really testing someone's white blood cells. We also have platelets that aid in blood clotting and the repair of damaged vessels. Some more composition of blood. Your blood also contains antibodies, which are proteins secreted by white blood cells that attach to antigens to destroy them. Antigens, which are foreign molecules or cells that react to antibodies. Enzymes, which are complex proteins that catalyze different biochemical reactions. And many enzymes and proteins have been found in blood that are important for identification purposes. And I bring this up because we have talked about how identical twins have identical DNA, but we are starting to find that the enzymes and proteins inside even identical twins are different and allow us to tell them apart. So everyone has a blood type. How do you get yours? So there are 4 major blood types and then 2 different antibodies. The 4 major blood types are A, B, AB, and O. And then you will carry the antibodies of either anti-A and or anti-B. So you get your blood type from your parents. You can determine the possible blood types for an individual if you know the blood types of their parents. So your parents are responsible for giving you your blood type. So your blood type is your phenotype. It is a physical characteristic that you display. But some blood types have multiple genotypes associated with them, different genetic ways that that blood type can be produced. And this is because blood types A and B are dominant over blood type O. O is a completely recessive blood type. But A and B are co-dominant with each other, meaning that neither of them is really stronger than the other, so to speak. And this is where our 4 blood types come from. If you have type A blood, your genotype could either be capital IA, meaning you are homozygous for type A blood, or capital IA, lowercase I, meaning that you are heterozygous for type A. And those individuals have anti-B antibodies in their blood because their blood is anti-B. It doesn't want any B proteins in there. Type B blood is the opposite, where you could have capital IB as your genotype, being homozygous for B blood, or capital IB, lowercase I, meaning that you are heterozygous, and you would have anti-A antibodies because your blood is anti-A proteins being in there. Blood type AB only has one genotype. You have to get one A gene from one parent and one B gene from the other parent, and you actually have no blood type antibodies. You might still have antibodies for like sickness and stuff like that, but no blood type antibodies because you have both A and B proteins in your blood. Last but not least, we have type O blood that you can only get by getting 2 recessive genes from each parent, so one lowercase I from each parent. And individuals with type O blood have both antibodies of anti-A and anti-B because they do not have any A proteins or any B proteins in their blood. Now, this does explain blood donation to us because you can only donate to an individual that you are not going to conflict with their antibodies. So O can give to anybody because there is no such thing as anti-O. So O can give to anybody. AB can only give to themselves because everybody else is going to have an anti-A or an anti-B that gets upset. A's can give to other A's or AB, and B's can give to other B's or AB, because that's going to be okay as well. Now you might be thinking, but I've heard them talk about blood types in terms of positive and negative. Where does that come in? So red blood cells carry proteins on their surface in addition to the anti-A and anti-B. And one of those is the rhesus factor, which is called the Rh factor. And this is a protein that is carried by some individuals. If a person carries the Rh factor, they are called Rh positive. If not, they are called Rh negative. So if you hear someone having O positive blood, that means they have type O blood and are positive for the Rh factor. Versus O negative blood has O blood but is negative for that Rh factor. Now Rh is a dominant trait, so only one copy of Rh positive is needed for a person to be Rh positive. So in the United States, 85% of individuals have a positive blood type, so a vast majority of our population. From there, type O is the most common type of blood, so therefore O positive being more common than O negative. And coming in very close second is type A blood. Then we experience a steep drop-off with type B blood being kind of uncommon and AB blood being very uncommon. Now, once again, going back to blood donation, this is pretty good for us since O can give to everybody. Having it be our most common blood type is definitely an advantage. Now, you'll notice this does say for the population in the United States, blood type percentages, unlike fingerprints, are not common across all different populations. In some populations, different blood types become more common than others, so this is only for the United States. Now you might be thinking, okay, but if blood type is class evidence, why do we even bother doing it? Why don't we just run DNA? And that is because blood typing is quicker and less expensive than DNA profiling. We're going to be doing it in class, and you're going to see how easy it is. It takes seconds for a forensic scientist to get a blood type. Meanwhile, DNA profiling could take hours to days depending on what needs to be done. So even though it produces class evidence, it could still help us link a suspect to a crime or even exclude a suspect, because if they don't have the blood type we're looking for, then that means we know they aren't a good suspect for us. So it can still be helpful even though it's only going to be class information. Now, are you ever going to go to trial with only blood type as your evidence? Absolutely not, but it's going to be a great starting point to save us some time and money up front. All right, that's all we're going to talk about today. In our next video, we'll dive more into blood spatter.